All right, you guys, today we're going to do the urinary system, our second to last lecture. We are almost done. So today we're going to be talking about the different organs in the urinary system, which is fairly straightforward. There aren't too many of them, but really we're going to be focusing most of the lecture on the kidney, since the kidney is really our location for urine production. And what is actually producing that urine is called a nephron. So we're going to talk about that unit of urine production called a nephron. And then we're going to go through those other kind of more simple organs of the urinary system and how we actually void the urine from the bladder. And then we're going to talk a little bit about our disorders and development at the end. So let's go through these organs of the urinary system. Pretty straightforward. We're probably familiar with some of these organs already. The bladder, right, of course, is part of the urinary system, as well as the kidneys. But we've got some tubes that are going to connect the kidneys to the bladder and the bladder to the outside world. So we have our kidneys, which connect to our ureters. And the ureters are what are going to carry that urine from the kidneys to the bladder. The bladder is going to store that urine for when we need to void the urine out the urethra. Now, don't get ureters and urethra confused. I know that they are very similar in their sound and spelling. So ureters, kidneys to bladder, and then bladder to the outside world is the urethra. So what are the main functions of the urinary system? Well, we are going to filter a ton of blood. So pretty much all the blood in your body is going to be filtered through the kidneys, and then we're going to resorb most of it, right? We're going to keep most of the water, the electrolytes, all that important stuff, but we're going to secrete all of the waste. So essentially it's just one big filtration system to get rid of mostly the metabolic wastes of the body. So your body and the cells of your body are constantly undergoing metabolism and producing waste. And so this is how we get rid of that waste. So we talked about the main function of the kidneys, which is filtration, right? So we're going to filter many, many liters of fluid out of the blood. So that we'll talk about in a little bit as well. But some other things is that it really maintains the chemical consistency of the blood as well. So it maintains, how much of the different electrolytes we have in the blood, as well as blood pressure. So how much fluid do we have in the blood, right? So we maintain a specific blood pressure, and the kidneys are really involved with the signaling and adjusting blood pressure as needed. And so because of all this filtration, we're going to excrete all of these toxins. So what are these different toxins that are produced, or metabolic wastes, I should say, that are produced by the body? So these are, like I said, these are produced by the cells, pretty much all the cells in the body, when they undergo metabolism. So urea is going to be produced when we break down proteins. So we're going to use all those amino acids, and we are going to produce urea. Okay. And then uric acid is another one, and it's a turnover of nucleic acids, which are the building blocks of DNA and RNA. Okay, so don't worry too much about that one, but creatinine is really the big one. So creatinine, it's also a good marker for kidney function. So you can measure the levels of creatinine, and if you have too much creatinine, then we know that the kidneys are not filtering it out and not excreting it, so we know that the function of the kidneys are decreased. But creatinine is produced during the production of ATP, and ATP is our cellular energy, right? So this is when the cells are doing metabolism, creating energy, so we're going to be also creating all of these wastes as well. and any other toxins too. So mostly we're trying to get rid of all these metabolic wastes, but say, you ingest or inhale some sort of toxin, then this is how we get rid of it as well. Not only does the liver do that, the liver is the big organ for getting rid of toxins, but the kidneys do as well. So let's talk about kind of the gross anatomy of the kidneys. So where do they live in the body? What do they look like? So just like some of these digestive organs, the kidneys live in the retroperitoneal space. So remember, they live just outside of that peritoneal cavity where most of the visceral organs live, right? But these guys start in the retroperitoneal cavity. They were never in the peritoneal cavity. They never had a mesentery, so these guys started there and are going to remain there, okay? And they live kind of right at the lower part of the rib cage. So the superior portion of the kidney is kind of protected by some of those thoracic ribs. But mostly they straddle the end of the thoracic cavity to the lumbar vertebrae. So don't worry too much about the size of the kidney. It's kind of about the size of your fist, maybe a little bit smaller. And the big thing is the location. So if you notice, and a little bit in the picture it kind of shows, is that the right kidney is a little bit lower than the left kidney. And why do you think that is? Think about maybe what lives in that right upper abdominal area. We talked about last lecture. That's right, the liver. So the liver lives over here, taking up a ton of space right under the diaphragm. So it's actually going to physically push that right kidney a little bit lower in the abdomen. So now let's continue with the external anatomy of the kidney. So very similar to the lungs, the kidneys have a region called the hilum, and it's on the medial kind of concave surface of the kidney. It's kind of shaped, like a kidney bean. That's why they're named kidney beans, right? So they have that kind of concave shape towards the medial aspect, and that's where all the vessels and nerves are going to enter and exit the kidney, just like we talked about the lung. So hilum is a very general term for this region where the vessels and nerves are going to be entering and exiting the organ. The kidney also has quite a lot of layers. So because it's not in the abdominal cavity per se, or at least that peritoneal cavity that we talked about, it's not as protected. So essentially it has a bunch of layers of connective tissue, of fat, to really help try to protect these kidneys in the retroperitoneal space. So first they have, a fibrous capsule, very dense capsule around the kidney tissue. And that helps from spreading disease. So say one kidney is affected, it'll keep it contained within the kidney itself and hopefully not spread it to the other kidney or anything else in that area. But the big thing is it has a huge fat capsule. So, and it's very species dependent on how much fat is around the kidney, but essentially it helps insulate it and protect the kidney. So very thick layer of, they calls it perirenal, so around the kidney, perirenal fat. And then around that, it even has another capsule, so a renal fascia, so a fascial plane of connective tissue, even around that fat pad. So lots and lots of layers to help insulate and protect those kidneys in the retroperitoneal space. So now let's look at the internal anatomy of the kidney. So if we take a sagittal section through the middle of the kidney, we see that we have an outer region and an inner region. So the outer region is called the renal cortex. Okay, so it's more superficial. It kind of looks a little granular in appearance, and in this picture it's kind of a lighter tan. So it really spans just from there to there. So that's the renal cortex. And then all on the inside is the renal medulla. So the renal medulla is made up of these kind of cone or pyramid shaped, kind of upside down pyramids called the renal pyramids. So the renal pyramids make up the medulla. So here's one renal pyramid. Here's another one. So see, they're kind of upside down, triangle shaped or cone shaped in 3D. They look like triangles in 2D, but in real time, they're actually, in 3-dimensional, they're cone shaped, okay? And then at the base or the bottom of those pyramids, it kind of comes to a point, and at that point, there's a bunch of these collecting ducts that we'll talk about, and they all come together into what's called these renal papilla. So we'll talk a little bit more about that in a minute. So each of these renal pyramids are going to drain the urine. So the urine is all going to be produced up in the cortex, up in that region, and we'll talk about that. And then it's going to drain out of the kidney, okay? So it has to go through a couple structures before it gets out. So it has to go through those renal papilla, okay? Then it's going to come into this kind of cup-shaped structure, and that is a calyx. Okay, so think calyx is a cup, and essentially it's going to funnel that newly produced urine into a minor calyx and then a major calyx, and then all of them pool into this large renal pelvis. So the renal pelvis is the true kind of middle area of the kidney, kind of in the hilum region. Okay, and then that's all going to funnel that urine out, that ureter. Okay, so each kidney has its own ureter that's going to go out of the kidney and to the bladder. So when we talk about the kidney, we have to talk about the vasculature, because the kidney is going to filter all of the blood, right? So we have to bring a lot of blood to the kidneys. So we have those renal arteries that are bringing in all of that freshly oxygenated blood into the kidneys. And then we're going to filter all of that blood. So how do we get to that filtration system of the kidney? Well, the renal artery is going to start branching into smaller and smaller arteries until we get into our capillary beds, okay? So first, we're coming in as that renal artery, and then we're going to branch as segmental arteries. So each one of these is a segmental artery coming off that renal artery. And then if you notice, it comes up and in between two of those renal pyramids, okay, that are making up that renal medulla. And that is called an interlobar artery. Okay, so if it goes through in between one of these lobules, so also these pyramids are called or referred to as lobules. So interlobar is going to be in between the lobules. So interlobar arteries are going to go up, and then if you notice, they're going to turn up and over the renal pyramid. So once it makes that turn, that sharp 90-degree turn, it becomes an arcuate artery. So think arch. Arcuate is meaning arching, so arcuate arteries. And then those guys are going to throw off even smaller little branches up into the cortex. Okay, so these guys are called cortical radiate arteries. So think they're radiating off the arcuate artery into the cortex. Okay, so that's why they're cortical. These guys you'll also see in lab manuals and things like that, maybe in the textbook, called interlobular arteries. So different than interlobar, interlobular arteries. So I like cortical radiate arteries better because it explains exactly where they are, but you will see the interlobular arteries in other textbooks and things. So don't get confused, they're the same thing. And then once they branch into these cortical radiate arteries, then we're going to get into the capillary beds. Okay, So that's where the filtration is going to happen, and we'll talk about this. So we have some arterioles that are going to go in and out of this structure called a glomerulus. So the glomerulus is where all that filtration is going to happen. Okay, so we have to push the blood into and out of this glomerulus, and the blood is going to filter out of the glomerulus. Okay, so let's talk about that. So before we talk about the filtration system, let's follow the blood through the kidney and finish it off. So we went through the arterial system, okay? So we are going to branch and we're going to get into these smaller and smaller arteries till we finished up at that cortical radiate artery. And then that's going to send off these afferent arterioles into the glomerulus. So that's where we said that's where the filtration is happening. So all the filtrate is going to now exit the glomerulus. Okay, and we'll talk about that region in a minute. But if we don't get filtered out, so if we're just, blood cells, some proteins, things like that that shouldn't be filtered out, that stay in the vessels, then we're going to exit that glomerulus as the efferent arteriole. So E for exit. Okay, and then we're going to actually go through the true capillary bed. So remember, that's where all the actual gas exchange is happening, the nutrient exchange is happening in that capillary bed. Okay, so that we'll talk about these capillary beds in a second. There's two different types of capillary beds that we could go through. We'll go through one or the other, depending on what type of nephron we are. And then we are going to exit the same way we came in, but through the veins, right? So now we're deoxygenated blood, but we're going to have the same named veins as we did the arteries. So then we're going to drain out those cortical radiate veins, which will then become arcuate veins, and then the interlobar and renal veins, all the way out to that inferior vena cava. So now we're going to talk about how we actually produce urine. So we bring in all that blood into the kidney from that renal artery, and we're going to filter that blood in the glomerulus. Okay, and so all of that filtrate that comes out will eventually turn into urine. Okay, so I have this great video for you guys, and I'll post it on Canvas as well, that shows kind of the how the nephron works and how much fluid your kidney actually filters in a day, which is a crazy amount of fluid. It's liters and liters of fluid, which is just crazy for these tiny little organs. So it's amazing what the kidneys can do. So we talked about these main functions of the kidney, right? So filtration is going to be kind of the number one thing that's going to happen. So we're going to filter all that blood. It's going to leave the glomerulus. So if we look at our picture over here, so here's that cortical radiate artery coming up, okay? Remember, we're in the cortex of the kidney, and we're going to shoot off that afferent arteriole into the glomerulus. So this little tuft or ball of arterioles is the glomerulus. So then the filtrate is going to come out. So we're going to be filtering all that blood, and some of it's going to go out, essentially the plasma, all the water, all the liquid, all that good stuff, all the electrolytes, everything is going to leave. Everything that can leave will leave. So essentially the only things that are going to stay are some proteins, some blood cells, things like that. Okay. And they are going to stay in the arteriole and exit out the efferent arteriole and go through the actual capillary bed, okay? Peritubular capillary bed. So that we'll talk a little bit more in a minute. But essentially, if you're a filtrate, so that's what we're going to focus on now. If you're filtered out of the glomerulus, you're going to go through the renal tubule, okay? So this is all part of the nephron, okay? So then we'll talk about all these different structures that we're going to go through in the tubule. So then we're going to be resorbed. So we don't want to lose all of that water and nutrients and ions in urine, right? We're going to just get rid of what we want to get rid of, and we're going to try to keep what we need and what we can. So most of the nutrients in water and all those essential ions or electrolytes, we're going to reclaim, we're going to resorb back into that capillary bed. Okay, and if we are something we don't want, such as a waste, we're going to secrete it. So we're going to actively secrete things out of the vasculature as well. So that's number 3. So again, we can leave as a filtrate. So we're going to try to get rid of the waste that way, but we can actively secrete things as well. Okay, and so that is what is going to end up finally as urine. Okay, so let's go through this process. So if we look at the nephron, and so the nephron is really the functional unit of the kidney. It is what is going to be producing that urine. Okay, so we have lots and lots of nephrons because, you know, we said we're filtering liters and liters of fluid in these kidneys per day. So we need millions of nephrons to actually do this. So that means we have about 1 million nephrons per kidney. So that's about 2 million nephrons. And they really only live in the cortex of the kidney. Okay, the medulla is really just collecting ducts and duct work. Okay. So what is the structure of the nephron? So we know the glomerulus. So we talked about the glomerulus, which is this tuft of arterioles, or blob of arterioles, if you want to talk about it that way. But we also, it is part of a renal corpuscle. So the larger structure here is a renal corpuscle. Okay. So the renal corpuscle contains the glomerulus, and then it also has a capsule. Okay, so the renal capsule surrounds the glomerulus, and both of those structures are part of this renal corpuscle. Think of it like a cup or a funnel. So it's going to catch all that filtrate that's coming out of the glomerulus. Okay. And then the other part of the nephron is the renal tubule. So we haven't talked about those different structures of the tubule yet. Okay. So let's first talk about the renal corpuscle. So it's the first part of the nephron, and this is really where the blood is getting filtered. Okay, so this is the site of glomerular filtration. Okay, so we have afferent arterial blood coming in. It's going to get filtered out of the glomerulus and go through the renal tubule. Okay, unless you're a blood cell, protein, things like that, and you stay in the arteriole and go out the efferent arteriole. Okay. So there are some important structures about the glomerulus. So how do we actually filter the blood, right? So how do we actually filter it? Okay. So the glomerulus is made up of a bunch of little capillaries. Okay, so these capillaries are fenestrated capillaries. So if we go back to our vasculature lecture, we talked about the different types of capillaries. We said the fenestrated capillaries are quite permeable. They're kind of middle of the range in the permeability. So these guys are going to be a little more permeable than most of our normal capillaries in our body. But they're not quite as permeable as those sinusoids, like we said, are in the liver and the bone marrow, things like that. Okay. And then that capsule, so it's called the glomerular capsule or Bowman's capsule. And think of this kind of like I said, like a cup, so it catches the filtrate, but also think of it a little bit like a serous membrane. Okay, because it has 2 layers very similar to a serous membrane. So we have a parietal layer, parietal outside, right, just like our serous membranes. So it's just this simple squamous epithelial outside. Okay, and then the visceral layer is up against the glomerulus itself, okay? Just like our visceral membrane and a serous membrane. So very similar. Think of it like that and you won't get confused. Now, the visceral membrane is essentially made up of these very specialized cells called podocytes, okay? So the podocytes have these little feet, okay? And we're going to blow up a picture of this to show you what it actually looks like. So podocytes, podo meaning foot, cell, site, right? So cells with feet. And they essentially are going to be surrounding the glomerulus, okay? So let's take a look at a bigger picture of that podocyte. So this is a really cool picture. So the top left corner, we have our original picture that we just looked at, right? Our renal corpuscle. So let's just blow up one of these little regions right here. And you can see these individual podocytes. So they have these little projections coming off of them, these little feet, okay? And they are going to be what are surrounding that fenestrated capillary. So the fenestrated capillaries has those fenestrations or pores, okay? So, and then between the two things, the fenestrated capillary and the podocytes, we're going to create what's called a filtration membrane, okay? Oop, I didn't quite circle that correctly. So filtration membrane, okay? So this is going to be made up of that fenestrated capillary, some basement membrane, okay, and those little foot processes of the podocyte, okay? And that is what is going to filter all this blood, okay? So essentially the plasma of the blood is going to go through this filtration membrane and out into that glomerular capsule, okay? So, and then this left picture is just really cool, actual picture of podocytes. So you can actually see the little processes, so they're kind of cool. So then I just have it all written down about this filtration membrane. So essentially, the filtration membrane is what is going to create the filtrate, which will then become urine, okay? So I just layered or wrote out the layers of this filtration membrane because I want you to know it, okay? So we have the fenestrated endothelium of that capillary, okay, which is just another word for epithelium, right? So fenestrated endothelium of that capillary, basement membrane, right? And then those filtration slits of the podocytes, those cells with feet, okay? And that's what's going to create the filtrate from the blood. Okay, so essentially what happens is, and why we have this filtration membrane, is so that we, filter what we want to come out. So we pretty much get rid of everything. So we try to let through water, ions, sugar, amino acids, but the big thing is that waste, right? So we want to get that urea out of here. But you hold back things that we want to keep for sure, like our blood cells and our proteins. We don't want to get rid of those. They're going to stay in the capillary, right? So they're going to stay in that, in the arterioles. Okay. So now if we're the filtrate, right, we've gone through glomerular filtration, we've left the glomerulus, and we're heading into the renal tubule. Okay, so this is where we're going to be resorbed if we're important, or we're going to be secreted if we are a waste. So what are the three sections of the renal tubule? So here, let's look at our picture, right? So here's our renal corpuscle. Okay, so we've been filtered out of that glomerulus and we're heading into the tubule. So this first section, which has a lot of turns, some squiggles and turns, is the proximal convoluted tubule. Okay, and now we're going to head into this big loop. We're going to take a dive into the medulla. Okay, so we come back up. So if you notice, we have our arcuate artery and vein here. And that is what is going to be the separation between cortex and medulla. Okay, so most of the nephron is in the cortex, and then sometimes we're going to take a dive into the medulla. And this is where all the concentrating of the urine happens, is in this nephron loop, or loop of Henle, a lot of people call it. Okay, doesn't have to go into the medulla, but sometimes it does, just depending on where it is in the cortex. And there are some that have very long loops of Henle that are really close to the medulla. We'll talk about those types of nephrons in a minute. But so if we go through our loop of Henle, we get concentrated, and then we're going to come back up and we'll go through another loop with lots of turns and twists, and that's the distal convoluted tubule. And then we're going to exit via the collecting duct. Okay, so the collecting duct is going to take a lot of urine from multiple nephrons. Okay, so multiple nephrons are going to all collect, why it's called the collecting duct, right? So it's going to collect all that urine into the collecting duct. Okay, and then all those collecting ducts are going to come together and be those renal papillae at the bottom of those pyramids. Okay. So now we know the collecting ducts are going to collect urine from multiple nephrons, and then it's going to drain it out into those cups, or those calyces, into the renal pelvis, out the ureter, and to the bladder. So essentially it's draining the urine from those nephrons, but at the same time it does have another important job. So essentially it's going to help concentrate urine as well. So we said the nephron loop is going to be really important for concentrating urine. So here's our nephron loop, but at the same time, so does the collecting duct. Okay. So remember when we were talking about the different hormonal controls of blood pressure and things like that? Well, this is where that ADH, or that antidiuretic hormone that's produced, secreted by the posterior pituitary gland. Remember, it's produced in the hypothalamus. But this is where it's going to act. So it's going to act upon the kidney, because remember before we just said, well, it causes the kidney to resorb more water. Well, this is where it is actually acting on. It's going to act on the collecting duct and the distal convoluted tubules, essentially increasing the permeability so that it will resorb more water from the filtrate. And that'll go into the bloodstream, which increases that blood volume, increasing that blood pressure. So now we said that there were a couple different types of nephrons. So more specifically, there are two classes of nephrons. So there's the type that we've been talking about, which is our cortical nephrons. And that's really 85% of those 2 million nephrons, right? So that is really the majority of the nephrons. But we do have this other type of nephron called a juxtamedullary nephron. So think juxta, or next to the medulla, medullary. So these guys sit right next to the medulla. So they're still in the cortex, but they're very low in the cortex. And this is only about 15% of the nephrons. But these guys are important because they are the ones that are going to be the most concentrating of the urine. So they are going to really concentrate the urine, even compared to those cortical nephrons. And how do they do that is because they have really long loops of Henle. And so these guys dive even deeper into the medulla. So that means that they're able to concentrate the urine even more, because we said that that loop of Henle is really important for concentrating the urine. Well, these guys are really even better at it. So in our picture here, we have our normal cortical nephron on the left. Okay, so these guys are the typical nephron of the kidney. Okay, so again, that loop of Henle is fairly short, but it does maybe go into the medulla, just depending on where they are. And then here is our juxtamedullary nephron on the right. So really long nephron loop or loop of Henle, and it goes very deep into the medulla and comes back. So that's going to even concentrate that urine even more. So now we've been talking about a lot of different types of vasculature within the kidney. And so the nephron has two different capillary beds. Okay, so the first type of capillary bed is the glomerular capillaries. So we already talked about these guys. So these guys are going to have the arterioles, the afferent arteriole coming in, becoming those glomerular capillaries, which are going to filter the blood and then leave as the efferent arteriole. And essentially this is creating a portal system. So a portal system just has to do with the same type of vessel. So again, we're not turning into the venous system after you leave the glomerular capillaries. You're going to continue as the arterial system. Okay. So that's a portal system. We've talked about the hepatic portal system, which is all venous blood. So essentially a portal system is a capillary bed, but you're not switching from arterial to venous. You're staying either arterial or venous, okay? And then we have our other type of capillaries, which are true capillary beds, right? So these guys are going to switch from arterial to venous. So we have either the peritubular capillaries, which are in those cortical nephrons, which is the majority of the nephrons in the kidney, or if we're talking about those juxtamedullary nephrons, which are our special nephrons that are just down by the medulla, they have a very special capillary system called the vasa recta. And essentially both of these capillary beds, either the peritubular capillaries or the vasa recta, these guys are going to come from that efferent arteriole. So the efferent is exiting the glomerulus, so that efferent arteriole is going to turn into either the peritubular capillaries or the vasa recta, just depending on which nephron you're talking about, okay? And this picture is just showing you those tufts of glomerular capillaries. So here's a glomerulus, which they're kind of cool looking, right? Like little clusters of vessels coming off of those arterioles. So we've already talked a bit about our glomerular capillary bed and how we're creating a portal system with those arterioles. We don't have any venous blood. This is all arterial blood, but we are filtering all that blood to create that filtrate, which will eventually become urine. But the important part about this system is that the afferent arteriole coming in is a lot bigger in diameter than the afferent arteriole coming out. So if you think about it, if that efferent arteriole, the exiting one, is a lot smaller in diameter, it's going to cause a backup of pressure in that glomerulus. So that increase in pressure, okay, so the higher pressure system in the glomerulus is actually going to push the blood through that filtration membrane. So remember we said we have our 3 layers in the filtration membrane. Well, how do you get the filtrate to cross the filtration membrane, right? You have to push the blood through, and that high pressure system in the glomerulus is how you're going to do that, which is caused by that smaller efferent arteriole. So now if we look at the difference between our peritubular capillaries, which are our true capillary bed in the cortical nephron compared to the vasa recta in the juxtamedullary nephrons, you can see a difference in where they are located and kind of what their job is. So the peritubular capillaries, that is in the name. They wrap around the renal tubule. So they're going to wrap themselves around the distal and proximal convoluted tubules. So essentially they're just wrapping themselves around the convoluted tubules. so that they can secrete and resorb all the water and ions and things that they want to keep, and then secrete all the waste that we don't want to keep. Okay, and then they're going to exit out the system, okay, out the venous system. So then we have our cortical radiate vein that's going to leave and become the arcuate vein, and essentially out the kidney into the venous system, right? So whereas the vasa recta is going to circle itself around the loop of Henle, the nephron loop. So it's not around the convoluted tubules, it's only around that very long loop of Henle. Okay, so this is how it's able to really concentrate. So it's really going to pull all that water and ions out of the filtrate, out of the renal tubules. Okay, so it's a little bit different on the location of where these capillary beds are, but they both arise from that efferent arteriole coming out of the glomerulus. Okay, so the efferent arteriole is what is going to turn into our capillary beds, whether that's peritubular capillaries or the vasa recta. Just depends on whether you are a cortical nephron or a juxtamedullary nephron. Okay. So now I've just written down all that information for you about these different capillaries. So the peritubular capillaries are the ones that are going to be wrapping themselves around those convoluted tubules, and they're only found in the cortical nephrons. So they are really for absorption. So they're going to absorb all the solutes and water that they want to keep, and then they're going to actively secrete any sort of waste that they don't want out of those capillaries into the tubules. Okay, so those are our peritubular capillaries. And again, all these guys are from the efferent arterioles in the glomerulus, leaving the glomerulus, I should say. And then here's our vasa recta, which is very similar to the peritubular capillaries, but they're just in the juxtamedullary nephrons. And the biggest difference is they surround the nephron loop instead of those convoluted tubules. So they're still going to be absorbing, so the function is very similar, but they're just going to even add to that concentrating ability of these juxtamedullary nephrons, which is why they are so much better, one of the reasons why they're so much better at concentrating urine. Okay. So now we have produced urine in the kidneys. So now we have to figure out how to get this urine out of the body, right? So the ureters are the main connecting system from the kidneys to the bladder. So what is the main function of the ureter? Is to really just drain the urine from that renal pelvis to the bladder. But the big thing is that the urine just doesn't run through the ureters. It is an active process. So there is a smooth muscle in the ureter which is causing a peristaltic wave. So essentially it's very similar to our intestines, right? So it's going to squeeze the urine from the kidney to the bladder. It's not just gravity alone, right? And so it's running from the kidney to the bladder, but how it enters the bladder is important. So it's going to enter kind of at the inferior posterior side of the bladder, and it's going to come in kind of at an oblique angle, which means it kind of comes in diagonally. And so that is going to help it from preventing backflow of urine up into the ureter. We want that urine to stay in the bladder so that we can void it and it will leave the body. So if we look at the histology of the ureter, what organ does it look really similar to? We may have just talked about it in the last lecture. It looks like the esophagus. Yep, it pretty much does. But the difference is that the mucosa of the ureter is transitional epithelium. Remember we talked about transitional epithelium as really being found in the urinary system? And what does it allow for? expansion. So it allows distension or expansion of the ureter as the urine is going through. So, and we have a very similar layering system as we do the GI tract. So we have 2 smooth muscle layers, okay, which is going to allow for that peristaltic movement. And then we have an outer layer of connective tissue, very similar to everything else, okay? And remember, we are outside the peritoneum. So we said that outer layer is called an adventitia when we're outside the peritoneal cavity. If we're inside the peritoneal cavity, like a lot of the digestive tract, then we are serosa. Okay, so very similar to the digestive system. And it looks kind of like the esophagus, okay, just with a different epithelium. So now that urine has made its way to the bladder, and pretty much the bladder is just this collapsible muscular sac. And so the big thing about the bladder is its ability to distend and expand. So essentially, it's really just storing the urine until you can go to the bathroom, right? So its ability to distend or expand is very good. So a full bladder, essentially it's going to become a spherical structure and expand up into the abdominal cavity. So it should sit kind of in the pelvic cavity when it's empty, but it really can expand into that abdominal cavity when it's full. It's outside the peritoneal cavity, but it can push up into that peritoneal abdominal cavity. And the location of it is, you can talk about it in association with other organs in the male and the female. And I have a picture of it, which will make more sense, I think. So essentially it sits above the prostate in the male, and it sits beneath the uterus in the female. Okay. So here's that picture of the difference where it sits in the male versus the female. So here's the bladder in the male, okay, and then here's the bladder in the female, okay. So here is the urethra coming out of both, okay, and then if you notice, the urethra goes right through the prostate gland in the male, and then the uterus sits right above the bladder in the female. Okay. So what are some features of the bladder that really allow it to distend? What are some anatomical features that are special about the bladder? Well, something that's not special, because we've seen it in other organs, such as the stomach, is rugae. So the wall of the bladder has folds in it, which allows for that distension or expansion. And then there is this region of the bladder called the trigone, and it's clinically important because it's where a lot of UTIs essentially stay. So infections will be persistent and they will stay in the trigone, because it can be a place where when you void your bladder, the little bit of urine can stay in that trigone area, which causes a persistent infection. But essentially the borders of it are going to be the entrances or the openings to the ureters, which are right here in the corners, and then the exit of the urethra at the bottom. Okay. And another feature of the bladder is a fetal remnant structure, just like we talked a bit about some fetal remnant structures in the heart. The urachus is something that used to be open in the fetus to allow for fetal waste excretion. Okay, so the urachus is this ligament, essentially here, that ties up to the umbilicus. So it used to be open in the fetus, right? So, the fetus is still producing waste, producing urine, but essentially where do you have it go? So essentially it's going to go out and into the umbilicus, so that the waste will go to the mom instead of, urinating in the amniotic sac. Okay, so there is some problem in some newborns called a patent urachus, where that it's still open, that urachus is still open and urine can come out of the umbilicus, but normally it should close off, you know, when the umbilicus closes off, and then it just becomes this remnant structure, kind of a ligamentous structure on the anterior side of the bladder. So I have a picture of the patent urachus. It happens in foals as well. So it's just where that urachus is still open. So urine is able to come out the urachus and out the umbilicus. So the picture on the left is a human baby, and then the picture on the right is a foal that has a patent urachus. So it's urinating out of the umbilicus as well, which I think is a little better of a picture to actually appreciate it than the human one. So now if we look at the histology of the bladder, the important thing is that epithelial layer is the transitional epithelium. So just like the ureter, we have the transitional epithelium to allow for that distension. So not only does the bladder have rugae, the folds in the wall of the bladder, but also this transitional epithelium to allow for that expansion. It also has a very thick muscular layer. So instead of a couple layers, it just has one thick layer called the detrusor muscle, because really the only function of the muscle is to squeeze all the contents out, right? It doesn't need to do the two different motions like the GI system. And then on the outside, we have that fibrous connective tissue, the adventitia. Okay, so very similar layers again as the other structures in the urinary system, as well as the gastrointestinal tract as well. So now the last part of the urinary system is the urethra. So that's how we are going to exit out of the body and out of the bladder. So we also switch epithelium in the urethra. So we're going to start out as that transitional epithelium near the bladder, and then we're going to switch to stratified squamous epithelium. And in between that, in the males, it does switch to the pseudo-stratified columnar, kind of mid-urethra, but in the females, it just kind of switches into that stratified squamous epithelium near the kind of distal end of the urethra. And there's a difference. So between males and females, there's a difference in the length of the urethra. So in the male, it's quite long, right? So it has to make its way all the way through the penis. So essentially it's about 20 centimeters. So it's quite long, and it has 3 different regions. So we actually name the different regions depending on kind of where it's going, what it's going through. So the prostatic urethra, it's going through the prostate. That's the first part of it. The intermediate urethra is going through the body wall, essentially. And then the spongy urethra is going through the penis. So 3 different sections in the male urethra. Versus the female urethra is really short. It's only about 3 or 4 centimeters. So essentially it just kind of goes through the body wall. Now you may think, okay, now what might be a problem with a really short urethra? What might come back up and cause problems? What do females get more than males? UTIs, right? So we can get UTIs a lot more frequently because that urethra is so short. The bladder is a lot more easily exposed to the outer environment. So now let's talk about micturition. So micturition is urination. How do we actually tell our bladder to urinate, right? So we have 2 different sphincters in the urethra. We have an internal urethral sphincter, which is smooth muscle. And really this is just kind of at the base of the bladder. So it's right at the opening. Okay, so right as it comes right out at the base of that trigone. And essentially it prevents the dribbling of urine, because that would be pretty awful if you just kind of constantly dribbled urine. That would be pretty bad. But you have no control over that, okay? Versus the external urethral sphincter is voluntary muscle. So it's really at the abdominal wall, okay? So it's at the abdominal wall. It's skeletal muscle, so you have control over it. But guess who does not have control over it? That's right, babies. So babies have what's called reflexive voiding, meaning that they don't have control over that and they just urinate whenever their bladder gets full enough. Okay, that's why when you change diapers, right, you might get urinated in the face. So babies have to learn how to control that external sphincter, right? And then hopefully we maintain that control. So the innervation is important. So it's all the autonomic nervous system when we're talking about that, the muscle, the detrusor muscle, as well as that internal sphincter. So we have control over the external sphincter, but the autonomic nervous system has control over the detrusor muscle and the internal sphincter. So the big thing I want you guys to know is that the sympathetic nervous system inhibits micturition, right? You don't really want to be urinating while you're running away from the tiger, right? Whereas parasympathetic empties the bladder or, promotes micturition, okay? So what does that look like, right? So sympathetic, you're going to relax the detrusor muscle. You don't want to be squeezing the bladder, and it closes that internal sphincter really tight, right? Whereas parasympathetic is going to contract the detrusor muscle and open the sphincter, okay? So that's the process of micturition. So you have to have a combination of the autonomic nervous system functioning correctly, as well as the voluntary control of that external sphincter. So now let's talk about some disorders of the urinary system. So we've touched a little bit on UTIs, or urinary tract infection. I don't think I actually said that all the way through, but most of us know what UTIs are. And we said these are really a lot more common in females, right? So because of that really short urethra, and it's opening to an environment that's close to contamination, such as E. coli or other bacteria. And so it can be very common in females. And you have a burning sensation when you urinate, right, during micturition. And unfortunately, some UTIs can develop into pyelonephritis. And essentially, pyelonephritis means infection of the kidney, but it usually is spread from a UTI. So from the bladder up the ureter into the kidney. So that's when you get into some life-threatening situations with pyelonephritis. You can also get kidney stones or renal calculi. And these are super painful. So you develop the kidney stones in the renal pelvis, okay? And then what happens is that they sometimes can go through the ureters, and that's when it gets really uncomfortable. Because like we said, the ureter isn't just a passive tube, it's actively squeezing that urine down into the bladder. So if it has a stone in there and it's trying to squeeze it, that is super painful. It's one of the most painful things I've heard for people that do develop renal calculi. Other animals as well develop renal calculi. Some cats and goats and things like that, and they can be just as painful, and sometimes they need surgical correction. You can do some shockwave therapy to try to break up the stones, but most of it is trying to manage your diet and increase water intake and things like that. Of course, we have cancer of the urinary system as well, specifically in the bladder, but also in the kidneys too, right? So those two are our big sites for cancer. So now we're going to finish up with a little bit of embryonic development of the kidneys. And the cool thing about this is that you actually develop 3 sets of kidneys, which is kind of crazy. So you have 2 of these kind of fetal kidneys that are going to either go away or turn into something else. And the first one is a pronephros. So the pronephros is essentially just going to kind of degenerate as the mesonephros comes in. So this picture on the right is showing this mesonephros. So this large structure here is the mesonephros, and then just that distal end, or the, whatever end it is going to be the degenerating pronephros. And then if you notice, there's a little bud coming down here, and that's going to turn into the metanephros. And that's the one that actually becomes the adult kidney. And you might be like, well, why is there other kidneys? What's that mesonephros? Well, little preview, this mesonephros is going to become your reproductive system. Okay, and it's going to develop into either male or female reproductive systems. So that's kind of cool. And then the metanephros stays around and becomes your adult kidney. And you're actually producing urine by 3 months in the fetal age, which is crazy. Okay. So as the urinary system ages, you do get quite a bit of decline in function in the kidney and the bladder. So essentially those nephrons kind of decrease in size and number, and the nephrons or the tubules are kind of less efficient at secreting and resorbing. So you definitely get this decline of urine production. So that can be a problem. As well as some nervous system problems. So you get, a lack of desire to urinate. So the feedback system isn't quite proper, the sensory system. And then you also get a loss of muscle tone in the bladder as well. So again, the joke is we start in diapers and we end in diapers, right? Unfortunately. So here's our learning objectives for this lecture, a little bit shorter than some of our other ones. And next time we're going to talk about the reproductive system. So that's going to be our last lecture. I can't believe we are almost at the end, you guys. Stay safe.